today my talk is about the introduction of a zebrafish screening platform system for clinical effects of natural products, including urban medicine and others. So first of all, I would like to start with some introduction about some animal model for biomedical research. So historically, for biomedical research, several animal models have been used for each different type of aspect of field of biology. So like for last whole century, for doing some genetics, fly and worm, those kind of invertebrate animal models has been used really, really widely. And for embryology, like developmental biology, the frog and chicken model was used very actively. But most of all, for human biology and clinical studies, the mouse system is the powerful genetic system and also very useful model, animal model for clinical science. And among those, I'm going to talk about the other model called like a zebrafish model system. So zebrafish is very interesting model because they are not the best one for each aspect of category like this. But the interesting thing is fish has everything, not kind of the best one, for it has some of genetic embryology and human biology for this. That's why I think this model is emerged as a kind of one of the alternative powerful model system from 1990s. And I'll give you a very brief introduction about this model system. And for zebrafish, they can, first of all, their development is very fast. So by 24 hours post-fertilization, and they just kind of made whole body plan like that. And also most organs start to form. And also they possess all types of like a vertebrate system hallmark, including tissues and organs. And actually they have a transparent embryos, which is optimal for imaging through the whole process of development. And that's why I think for embryonic process study, it's very practical. So including this type of advantage, why zebrafish for kind of biomedical research? And the first thing is it's genetic similarity with human. It's almost like 10 years ago when actually bioinformatics study just kind of revolutionizing all types of system biology. And scientists found like each different types of model system has their unique genome information. And very interestingly, in zebrafish, 80% of disease-involved human genes are conserved in zebrafish too. That's when actually the whole field is excited to use zebrafish as a model system for like a human research and biomedical research. And next thing is when you're doing some kind of genetic work, the kind of first thing you have to do is genetic manipulation. To kind of find some clue for genes function, you need to do several types of genetic manipulation. And in fish, you can do a lot of different types of genetic manipulation is available in fish system. First of all, like a random mutagenesis for forward screening. And actually kind of, it's really strong, really powerful tools like a genome editing, like CRISPR mutagenesis. And also transient knockdown approach is very practical in fish. That means like you can do whatever you want for genetic manipulation part using this model system. And finally, due to their kind of optimal size and the number of embryos we can have from like a single, mating from one single parent, drug discovery platform like for chemical and drug screening is very available. Because one of the key thing is when we're using like a 96 well plates for kind of regular experiment, zebrafish embryo, like a larvae, has a really perfect size for each feet of this well plates. So that's why you can do different types of screening in a really large scale. So combine all those distinct types of advantages of zebrafish model system. Based on that, we can do and we can use this system for really drug discovery screening work using zebrafish model system. And to develop some possible candidate for drugs, some specific disease like that. And these are not just kind of, hypothesis or kind of research-driven work. So currently, like in the United States and also, the European group developed several drug candidates based on this large-scale screening using zebrafish model system. This is kind of single report, like already like 4 years ago in 2019 in United States group like Harvard and Utah University and any other university just kind of found several candidate molecules for distinct types of disease. And some of them are kind of enter early clinical trials. And this report is already 4 years ago. So much more candidates has been found from this screening work, and people are developing this kind of drug candidate for further usage. So that means like, using this fish model system is not just kind of idea, kind of our fantasy for now. So mainly based on this kind of introduction, I would like to give you some introduction about this zebrafish model system with importance of zebrafish as a in vivo drug screening tool. And there will be 4 main distinct kind of object for this talk. And first one is I'm going to give you what's the meaning of zebrafish as a bridge between in vitro and in vivo system. And also, zebrafish can be a good alternative model system because it can provide 3 Rs, which means like some ethical issue. For ethical issue, they can give some good example to provide 3 Rs value to drug discovery. And also, they can be a good preclinical evaluation for toxicity. And finally, I'll give you using some of the efficient disease model of zebrafish to do some drug screening for developing drugs. So first of all, fish, like, I hope you can understand because I'm here to selling this fish for you guys. So the first one is the fish is a kind of good bridge between in vitro and in vivo system. When you're trying to develop some drug candidate, you're going to need several model system to develop the toxicity and effect of those drugs, including like a cell culture system, and more importantly, like one animal system, like a mouse model system. And each system has their advantages and limitations. And as you can see here, zebrafish has some of advantage like a high reproductive rate, and development is external and fast, and genetic similarity is there. And they are also organism. That's the big thing. They are vertebrate organism compared to like a cell culture system. Still, there's some limitations there. But the meaning of a bridge between cell system and mouse system is about efficiency. You can do kind of finding some, especially when you're doing some large scale type of screening, it's kind of really efficient to use those type of like a vertebrate animal system directly for screening of those drugs. So they can just kind of make some bridge for that. And already 2 years ago, Randy Peterson group just kind of make very interesting review paper comparing this model system with some efficiency of usage and some possible usage for like a human patient disease model system. For example, when they make like when they evaluate those systems based on human relevance and phenotype relevance and some tractability, which means like ease of screening experiments, they got like a definitely human patient themselves should be the ideal model to do studies, but it's impossible to do screening experiment. That's why they score like a 0 for those. But as you saw here, like in cell culture system, the phenotypic wise, it's really tough because the cell itself is homogeneous cell level, which is inside of the cellular system. So sometimes it's tough to get some phenotype from the screening. And, like a high technology, like a iPSStem, induced pluripotent stem cell, or like organoid study, they are really ideal for some specific, the patient analysis. That's kind of really strong one. But for the screening experiment wise, it's really limited. And interesting here is zebrafish and rodent model like a mouse. They are also really great genetic model system. But one advantage of zebrafish is they are really practical for screening experiments. So based on this, they can just link this cell system to rodent system with really good tractability of screening tools. And these advantages can be summarized like a feasibility of generation of genetic model of disease. And also they got potential for larger screening platform here. And also drug treatment is very practical using these aquatic animals through this kind of the immersion of each drug candidate to see the effect of them. And second thing is zebrafish can provide 3 R's value to drug discovery. So basic research level and also clinical level and also kind of commercial level, the animal model system has been used widely. But recently there are kind of, strict regulation is just made to prevent unnecessary usage of overusing of this animal system. It's kind of really important ethical issue. And they got like a 3R in animal research with the replace, reduce, and refine. Replace means like, if possible, replacing this animal model system to other alternative model system. And second, reduce is just do not use unnecessary number of animals. So just use the kind of the minimum number if possible. And finally, refine means like reduce the possible suffering or stress of those animals for kind of usage of this animal system. And for 3 R's, it's kind of ethical issue at the same time. And we have a very standard regulation for now, especially like a commercial usage. It's kind of almost impossible to use rodent animal for now. So, but interesting here is from the European Commission directive from 2010, fish can be a good alternative animals based on this regulation. Before 5 days post-foliation, zebrafish can be used for lots of screening platform work too. That's why, and also as I just kind of give introduction from previous slides, like a zebrafish can make all types of organ and tissues from like a 24 to 48 hour post-fertilization. That larvae stage is also very practical for all types of physiology and also biology too. So the main reason why we can say zebrafish can provide this 3 R's value is here. First, for replacement part, when you're using some relevant model of disease of zebrafish at larvae stage, we can replace this animal toxicity study. For toxicity study, we can replace this one for others. And second thing is reduction means zebrafish larvae can be used for toxicity drug candidate studies, and it can allow safer molecules to be tested the higher vertebrate like a rodent system. And finally, for refinement, because the zebrafish embryos are transparent and we can monitor all types of development from like a fertilization to whole developmental process, we can allow for non-invasive observation of toxicity and possible recovery too. So this zebrafish can become like some good alternative models for, especially for rodent model based on this 3R value. And third part of zebrafish, usage of zebrafish as a kind of efficient tool is it can provide preclinical evaluation drug toxicity very well. As you know, like a toxicity is the key thing, especially in preclinical level to develop some drug candidate for disease. And in zebrafish field, especially this statistics shows, the report shows like a usage of zebrafish trend is getting increased for the next several decades. So early 2007, compared to like 2007, in 2018, you can see the publication number of zebrafish itself is more than doubled up. But more interesting thing here is because zebrafish has been developed at first for genetic model to study developmental biology and genetics. But toxicity was not the major one. But from like early 2007, some point, like people are trying to use this one for toxicity study to develop drugs like that. And it continuously increased, those tendency was increased like this. And Good thing about this zebrafish is we can use it for definitely toxicity studies. But as you know, when you're using a cellular system for toxicity studies, it's all about like a cellular senescence or a cellular, death. Those type of very kind of one-dimensional phenotype is the key thing. But zebrafish's kind of main advantages like mouse, this is organism. So when you're checking like a phenotype, first thing is definitely developmental phenotype, including like a lethality or survival rate type of thing. But zebrafish has all types of organs like a very early stage. That's why liver, like a nerve, gut, blood, and heart, and all tissue-specific phenotype and tissue-specific toxicity can be monitored using this model system. This is kind of another meaning of the bridge between cell and mouse system, because of the direct screening using zebrafish can give you information about this type of tissue-specific effect too. So I'm going to give you some of the examples we're doing in our lab currently. So we'll check some toxicity response with some herbal medicine. Actually, when I made like title with the herbal medicine, but there's no herbal thing here, but because we use like a bee venom and deer antler extract and hominis placenta to check some toxicity with this model system. And very interestingly, each those drugs candidate and natural product has some different lethal effect on zebrafish model system with different level of concentration like this. So this one is kind of one example to show those drugs effect on developmental process or kind of animal survival itself. But more interestingly, they can give some good information about tissue-specific wise. These are kind of one information is like a bone formation. So when you're using bee venom and deer antler with this zebrafish larvae system, you can see in some specific dosage, you can see the bone formation is kind of robustly reduced, which means like we can use this one to see the toxicity in some tissue-specific manner. This case is kind of bone formation, but also you can see like a blood formation too. So in zebrafish, we have several markers to label some specific tissue, in this case, blood formation. So here, MPX and L-plastine is well-known markers for myeloid among the blood cells. And when you're treating deer antler extract or placenta, you can see here like a really robust increase the number of these MPX positive cells compared to control, as you can see here. So this part is kind of that tail part of zebrafish where actually blood cells are formed and also proliferate to be functional. And as you saw here, like those increase of the neutrophil, because MPX and L-plastine is a major marker for myeloid, including macrophage and myeloid neutrophil cells. And not just only the kind of, blood formation phenotype itself, the increase of those neutrophils means like inflammation is also induced. So those type of specific tissue-specific effect can be monitored using zebrafish, including blood and bone. So those are the example of using this model system for toxicity trial. And finally, I'm going to give you some of the usage of zebrafish for direct drug screening effect. So key thing about zebrafish is due to the size, we can do several chemical screening within this type of 96 well plates. But more important thing is, as I told you, genetic manipulation is really practical in fish model. That's why we can make several types of disease model. This is one well-known example in zebrafish field. This is skin cancer model, melanoma model. As you can see here, when they're making some transgenic within some tumor suppressor gene mutant background, this type of melanoma formation is really well activated. This is kind of one good example of the disease. Using those type of disease model and large scale screening can be performed to find some possible candidate for those specific disease model. And in our case, we're making not just like a genetic model, but also you can generate some models using some injury induction. In our case, we developed some inflammation induction model to find some possible anti-inflammatory response drugs for further studies. And for recently in zebrafish field, the people are trying to generate to make some suitable and optimal model for inflammation induction. And they're using several types of drugs and chemicals or direct injury case like a tail transection, because the zebrafish has really high regenerative ability, and those kind of the injection or transection or injury model is also kind of widely used. And through this kind of induction of inflammation, we can also monitor those kind of effect using several readout of inflammation response. As you saw here, like a macrophage and neutrophil, interleukin-1b and TNF-alpha. Those are kind of good markers for myeloid, especially, which means like inflammation induction. And also using like a Sudan black B staining or MPX marker with in situ hybridization, you can see the kind of marker for inflammation. Those dots are kind of boring dots, but that's the kind of good indicator for inflammation. So using these tools, we're trying to set up our inflammation induction model, and we chose LPS treatment, this lipopolysaccharide, which is usually kind of induce the inflammation in the system of cell or animals. And we successfully induced the number of the Sudan black B positive cell, which is neutrophil. And also we're using dexamethasone, which is well known anti-inflammatory kind of drugs, to do some positive experiment to see whether this system can be used or not. And as you can see here with this quantification, when we treat it with LPS, those inflammation is induced very successfully. And co-treatment with this dexamethasone and LPS, we can see the kind of reduction and alleviation of those inflammation successfully, which means like this system is working fine. And next, we're continuing our work in collaboration with the Aju University group. And in this case, they just got some library of natural products, plants from Costa Rica and Nicaragua, and they're trying to find some anti-inflammatory effect from that plants. And what they did was first screening using cell level, and they tried to find like some possible anti-inflammatory effect. This is kind of one good example to show about like a role of bridge of zebrafish from cell to mouse system. And first screening is done from Aju University, and they did some cell screening successfully and found like some of a good candidate which has some potential to give some effect on anti-inflammation. And using this natural product, we performed like a similar experiment using our system. And when you try this candidate product called like a Guarea microcarpa, and as you saw here, LPS induced inflammation is robustly reduced with the treatment of this Guarea product. And also the other candidate, Ioea pardiformis, that treatment can also reduce those kind of inflammation induced in zebrafish model system using LPS treatment, which shows like a very consistent effect in cellular and animal model to develop some possible natural product effect. So that's the whole thing I got today for this morning session. And here's some summary for this, because mainly our talk is mainly about to give you an introduction about this zebrafish model system for drug screening. So for chemical screening, zebrafish plays a good bridge role. And also for ethical issue, they can give some good information about this 3R value for further work. And also zebrafish can be useful for evaluation of drug toxicity, especially in tissue-specific manner. And finally, using some relevant disease model system, we can use this system with really diverse drug screening platform. And finally, we're also trying to develop several other types of the screening system. And one example is in collaboration with this UNIST group, we're trying to make like an automated high throughput screening system, because the key thing about large scale screening is if we have more data and more number of screening data, the data and the result is much more powerful. To do that, we need to make everything automated like this. So through the automated system, we're using the general scanner system, and together with some AI and deep learning technique, we find like automatic phenotyping tools. And this is one example. And when we got this type of raw data, this AI system can kind of recognize the mutant and wild type very successfully with more than 99% accuracy. That means like we can develop and we can just use this model system for further screening work based on this automatic screening like this. And we hope this one is just mainly about the phenotype screening, but we hope we can just kind of expand this knowledge to further disease model system. So that's it for today. I would like to thank to our lab member, and also especially for our collaborators at Kyung University, the first speaker, Kyungju Park, and the old professor, Yeoncheol Park, Professor Byung-Kwan Seo, and Professor Yong-Hyun back here. So, and also I would like to thank to our collaborator at Aju University at UNIST. Thank you.